So good afternoon, everyone. Um, Welcome to the endocrinology grand rounds for today. We are very thrilled to have a visiting professor come here today from Chicago. Um, Dr. Sargis is MD PhD, currently an associate professor at the Department of Medicine in the Division of Endocrinology at University of Illinois in Chicago. He is also the uh, section chief of endocrinology at the VA, the Jesse Brown VA Medical Center. Dr. Sargis is a physician scientist with a interest in how the environment may contribute to obesity and diabetes. He obtained his BA in chemistry from Carleton College and then went on to do his MD and PhD at the University of Chicago. He did his residency in internal medicine and endocrinology fellowship at the University of Illinois. And then he joined the faculty at the University of Chicago in 2010 and was there for about almost 10 years and moved to University of Illinois in Chicago, where he has been since then. He's an outstanding clinician, educator, and researcher. His clinical practice is mainly in diabetes and endocrinology and lipid disorders. He has groundbreaking research which focuses on understanding how humans alter the environment and how these alterations promote development of metabolic disorders such as diabetes, obesity, and NASH. His research is funded by the NIH, the VA, the Department of Defense, and other funding agencies. And in addition to being a very active researcher and publishing his papers in different journals, he's also been involved in trying to bring public awareness of this problem and also to affect policy changes through collaborations with other organizations. So his eventual goal is to leverage the sound science to improve clinical and policy efforts to address the deleterious effects of environmental pollution on human metabolic health and its disproportionate impact on vulnerable populations. So he'll be touching on one of these topics today, as you can see from the title, environmental toxicants, health justice, and metabolic disease pandemic.
I just want to thank Sri for such an amazing introduction and the invitation to be here. I've had an awesome visit so far, meeting with folks, great conversations, and really look forward to continuing these interactions and as well as meeting some new folks tomorrow as well. So just by way of disclosure, I do some consulting work for CVS Health. It has nothing to do with what I'm going to talk about today. But I should say that I am here solely in my capacity as a faculty member of the University of Illinois Chicago, and that the views expressed are my own and not necessarily reflect positions or policies of the Department of Veterans Affairs or the United States government. So I just want to make that abundantly clear because of what I'm about to talk about. In addition, I just do want to mention that I am engaged in some community activities that I think are interesting. One of them is this Naperville Environment and Sustainability Task Force, which actually has a relationship to Kentucky, which we can talk about in the Q&A section at the end, which I mentioned to a couple of folks, as does the Clean Energy Alliance of Naperville. Been involved with the Food Packaging Forum a little bit, as well as a couple of Endocrine Society activities related to both endocrine disrupting chemicals and increasingly a new group that's formed related to climate change as well. So, but all of that really is informed by the mission of my lab, which Sri, I think, articulated pretty well, which is to understand how humans alter the environment and how those alterations impact human health. And critically, as a physician scientist, I'm not just interested in understanding how these chemicals cause problems, but I'm interested in translating that knowledge into action to protect the metabolic health of our patients. And when I think about our patients, I'm often reminded of Fannie Lou Hamer, who gave this famous speech in New York in 1964. She was a famous women's civil rights and voting rights activist who gave this speech in the presence of Malcolm X, in which she famously said, "I am sick and tired of being sick and tired." And when I think about this, I think about the patients that I care for in clinic and all those things that drive their disease that are out of their control, that they don't know about, and that are adversely affecting them and their family members. And it reminds me that our job is to protect them. And as physicians, we need to be sick and tired of the suffering that we witness, and we need to get off our butts and do stuff about it. So that informs, I think, a lot of what I do. So what we're going to talk about this evening fits within that context. And what we're talking about are these endocrine disrupting chemicals. So endocrine disrupting chemicals are defined by the Endocrine Society as exogenous chemicals or mixtures of chemicals that interfere with any aspect of hormone action. So traditionally, these are chemicals that are thought to disrupt things like estrogen signaling, androgen signaling, or thyroid hormone signaling. But over the course of the last 15 years, they've been increasingly associated with conditions such as obesity and diabetes, which are the areas that I'm most interested in. But what's important is that these chemicals are also associated with diseases that we won't talk about. These same chemicals are linked to cancer, psychiatric disorders, neurocognitive impairments. So for the things that I'm going to talk about today, I'm going to give you my perspective through the lens of metabolic disease, but just know that these chemicals are also important for other diseases of significant public health impact. So probably for this audience, I don't need to do this, but just to make sure that we're on the same page, I just want to make clear that when we talk about diabetes, what we're talking about is a condition that is defined phenotypically by elevations in blood glucose levels. Because this condition is defined phenotypically, it can be many different diseases that ultimately present with that final phenotypic output, which is hyperglycemia. And we know this inherently in diabetes because of the differences between type 2 diabetes, which accounts for 90 to 95% of the disease associated with sleep disruptions, physical inactivity, and obesity, and contrast that with type 1 diabetes, which is the autoimmune destruction of the pancreatic beta cells and the loss of the capacity to make insulin. There are also monogenic forms of diabetes, gestational forms of diabetes that affect up to 14% of pregnancies globally, as well as other forms as well. At their core, though, each of these diseases arise from a mismatch between insulin secretion and insulin action, which is shown here on the right panel. So for those who have normal blood sugar control and don't have diabetes, our insulin secretion is matched to our insulin sensitivity. So if we find a religion, we start exercising, we lose some weight, we become progressively more insulin sensitive, and our body makes less insulin to keep blood sugars in that tight range. The problem, of course, is that society is moving in the opposite range. We're gaining weight, we're becoming less physically active, we have sleep disruptions, and we become progressively more insulin resistant as a consequence. And our body has to make more insulin to keep those blood sugars in that tight range. The problem is that this stress this metabolic stress impairs beta cell function. They begin to fail, first under periods of peak demand, and one develops what's known as impaired glucose tolerance. And ultimately, those beta cells fail even during periods of fasting, and you develop frank type 2 diabetes. So when we think about the drivers of diabetes development, at their core, they're factors that decrease insulin secretion that may be a result of increased insulin resistance. And I'm going to come back to this in a minute, which will show you why I brought it up. So for a long time, what has been known about what drives the development of diabetes has focused on lifestyle factors, sedentary lifestyles, and dietary factors. But what we know is that these factors alone are insufficient to explain the magnitude and rapidity with which diabetes rates have increased around the globe. And so many years ago now, I was sitting at home one day and we put this slide together that basically shows a couple of interesting phenomena. So what you see in red here is diabetes prevalence in the United States from 1958 to 2008. And what you can see is that there's been this exponential increase in disease rates. And this has been mirrored around the globe, right? And it has continued to this day. So this data is a little outdated at this point, but this trend has continued. What's shown in blue is actually synthetic chemical production in the United States from 1938 to 1958. And what you can see when you look at these two curves together is that they are increasing together. And this is not suggesting that this is necessarily evidence of causation, but what this is evidence of is potential relationships that we now try to study to really establish that level of causality that we need to basically say that these environmental changes that we're seeing are causally related to the development of metabolic disease in individuals in the United States and around the globe. And so that's been sort of the mission of the lab since then, which has basically been to try to take this evidence into mechanistic studies to try to understand how they're related. And importantly, what we're talking about here is a variety of different chemicals. These are chemicals that are used in food production, that are generated through industrial activity, that we choose to use, chemicals in personal care products and in home care, as well as something that kind of sticks in my craw, especially chemicals that we use in the provision of medicine. So these are chemicals in medical products and equipment that we'll talk about a little bit later as well. So there's a million things, not a million, but there's many things here that we can study. And the question becomes like, where's the evidence that actually links these exposures to the development of diabetes? And so it turns out that that evidence is actually quite old. So there's evidence from the 1940s showing that alloxan, a chemical used in the production of a purple dye, is sufficient to drive the development of diabetes in rat models. And that's from 1943. There's evidence in humans that dates to the late 1970s when in a series of case reports, it was shown that individuals who attempted to commit suicide by consuming the rodenticide Vacor actually developed diabetic ketoacidosis. And we know that diabetic ketoacidosis is a condition that develops from a specific destruction of those pancreatic beta cells. This is the inability to make insulin as a result of Vacor-mediated destruction of those pancreatic beta cells. And since then, an increasing number of chemicals have been linked to disruptions in glucose homeostasis. So these are chemicals that are linked to impairments in insulin secretion, as well as impairments in insulin action. And these include a variety of things. So chemicals of both organic and inorganic origin, both that are natural as well as synthetic. And I'm going to give you some data from our lab that basically talks about two that I think illustrate the differences in which these chemicals can lead to a diabetic-like phenotype. So we'll talk about talafluinide, which we studied for a long time, that looks like it induces a type 2 diabetes-like phenotype, as well as arsenic that induces a more type 1 beta cell deficiency type phenotype. But what's important is like when we look at the diversity and consistency of the data, it really does suggest that toxicant exposures are novel diabetes risk factors that have been inadequately sort of taken into account in the practice of medicine. And so this is kind of depressing, right? So when you think about all of the things we're exposed to, this is particularly disturbing because we already can't get control of the risk factors that we already know exist. And now there are all of these additional risks. The thing with this, though, is I would argue that this is actually a unique opportunity because unlike genetics, environments are modifiable. And as a result, if we can make better choices about the environments we live and the chemicals to which we're exposed, we have the opportunity to bend this curve in ways that I don't think we've fully recognized in the past. And that's the argument that I'm going to make with. So where did this all start? So this all started for me with this fungicide called talafluinide. Talafluinide is a phenylsulfonamide fungicide used on fruit crops in Australia, New Zealand, China, India. Historically was used in the European Union as well. It was also used as a booster biocide in marine paints. So ships get painted with these copper-based paints that they impregnate chemicals into in order to kill off barnacles and algae in order to improve hydrodynamics. And the important thing here is that these chemicals are not covalently bound and they leach into water. And as a consequence, they get into the seafood supply. So while in the United States, talafluinide is not used, it was allowed on imported crops. And the imported crop with the highest tolerance limit was hops with a tolerance limit of 30 parts per million. So for the studies I'm going to show you, we did animal models looking at the effect of toluflunate addition to the diet at a concentration of about 65 parts per million, which is the equivalent if these mice just ate hops all the time. And maybe something for you to think about if the next time you're going to have your IPA. But we basically did studies using C57 black 6 mice and asked if we add this fungicide to the diet, what does it do to the metabolism of these mice? And what we found is in the upper left that after about a month, those mice that were on the fungicide containing diet gained more weight. And when we looked either at necropsy or when we did DEXA scanning, what we saw was that that weight gain was actually adipose tissue. They were gaining more fat mass specifically. There was no change in lean body mass. And then when we assessed like glucose and insulin homeostasis in these mice, we did glucose tolerance tests, give them a slug of glucose, see how their glucose responds over time in the same way that you would do a glucose tolerance test in humans. We can see that there's a marginal increase in glucose levels over time. They get higher, they stay higher longer. There's a higher area under the curve for glucose over time. This in a mouse is basically the definition of diabetes as best as we can assess it. And what's important is that when we then tried to assess whether these mice were what their insulin sensitivity was, you give them a slug of insulin, look to see how much their blood sugars drop. What we found was that their blood sugars dropped less. They stayed higher longer, which is basically the definition of insulin resistance. And collectively, this phenotype looks like what we might consider type 2 diabetes like. Glucose intolerance associated with weight gain, that's adipose tissue associated with insulin resistance. In addition, these mice had interesting changes in their feeding habits. They had disruptions in their circadian rhythm. They ate more food when they were supposed to be fasting, which we also know is metabolically deleterious. This fits within a realm, like we studied this chemical to death. I think we're the only people who are actually ever interested in this chemical, but it served as an important point. And what we were able to show is that this fungicide functioned like an environmental glucocorticoid. So it could activate glucocorticoid receptor signaling in vitro. It promoted adipocyte differentiation in 3T3L1 cells. We could show that it could induce insulin resistance in fat, specifically by downregulating insulin-related substrate 1, insulin receptor substrate 1 in adipose tissue. And through a variety of studies, we were able to show that it caused an increase in glucocorticoid receptor signaling in adipose tissue, which we think is the mechanism by which this chemical actually causes insulin resistance. And those disruptions in circadian rhythm that I told you about are consistent with what we see happens to patients when we put them on glucocorticoids. They get sort of disruptions in their circadian rhythm that were consistent. But as I said, I think I'm the only person that's ever been interested in this chemical. And so we were interested in actually moving the lab to things that were probably of greater public health significance. And for this reason is why we started studying arsenic. So arsenic is a metalloid that exists at various forms in nature, including different oxidation states, inorganic forms, and organic species. It's a natural contaminant, but has historically been used as a pesticide, with some arsenicals still used in pigs and poultry. It's also found in various agricultural crops, including rice, as well as seafood, although seafood's often an organic form of it. And as someone who practices at the VA, one of the things that really stood out to me is the fact that arsenic-containing pesticides were used during the Vietnam War. So you may be familiar with Agent Orange, which I think people have some working knowledge of, this herbicide that was sprayed during the Vietnam War. But Agent Orange was one of several rainbow pesticides that were used in Vietnam. Agent Blue was a mixture of two different arsenic-containing pesticides. And what's interesting about this is that 4.3 million liters of the stuff were sprayed in Vietnam by hand by US military personnel. And there's some areas of Vietnam near former US military bases that have arsenic levels comparable to levels seen in Bangladesh. So knowing that there's high levels that may be related to this. Additional sources of exposure include things like coal ash, forest fires, as well as some fire suppressants. And the reason that I think this is interesting is that there's a very clear intersection now between arsenic exposure and climate change, another threat that we've become interested in. And then the main reason that we study this is that the estimates are currently that arsenic contaminates the drinking water or the groundwater underneath somewhere between 94 and 220 million people around the globe. So unlike talifluinid, this is a chemical of immense public health significance. So we were interested in understanding what this chemical was doing from a glucose metabolism standpoint, from a diabetes risk standpoint. And what we saw here is what the upper left panel is meant to show you is that we provided the arsenic in the drinking water at a pretty high level, 50 parts per million. But what we got were liver arsenic levels that were comparable to highly exposed populations in Bangladesh. Those mice exposed to arsenic through their drinking water had no change in body weight. There was no change in body composition. They went along the same track as the control mice. But what we did see was that those mice developed pretty marked glucose intolerance. When you gave these mice a slug of glucose, their blood sugars got higher and they stayed higher longer. Again, the definition of diabetes in a mouse. This was not due to insulin resistance in this model, but rather this was due to an impairment in insulin secretion. And what's shown here is just the delta in insulin secretion from 0 to 15 minutes, or a loss of first phase insulin secretion. There are multiple mechanisms that have been proposed to explain this decrease in insulin secretion. We've done some studies showing that this can occur specifically through a downregulation in serotonin signaling in the beta cells that may also relate to some of arsenic's toxicity in other tissues. So what we see in this picture is something that looks a little bit more type 1-like. So not the sort of immune component of this, but the decreased insulin secretion component of this. So we wanted to sort of increase the relevance of the things we were studying. And honestly, you're only interested in mouse diabetes as a model of human disease. So one of the questions was, is it possible, that we can take this into human studies as well? And so we did that. We had this fortune to work with Craig Hannes, who's been studying diabetes in Starr County, Texas, for over 40 years now. So Starr County is the third county up from the tip of Texas. And if you look at USGS maps of groundwater arsenic, what you can see is that it's a relative hotspot in the United States for arsenic levels. And so Craig and I had been talking for a while about, when we were studying talifluinid, there was no reason to work together. But when we started moving into the realm of arsenic, this was a unique opportunity for us to study the impact of arsenic on diabetes risk in a human population. And so, and we did this in collaboration with Maria Argos, who's at the School of Public Health at UIC and has since moved to Boston University, as well as my MD Ph.D. student, Maggie Weiss. And so the reason Starr County is a great place to study this is that diabetes in Starr County really matters. So Starr County is basically 100% Mexican American and has a very high poverty rate. So you have almost 40% of folks living under the poverty line in Starr County, and it has an astronomically high risk of diabetes. So if you're born in Starr County, you've got a 70% lifetime risk of developing diabetes. And importantly, it has among the highest rates of diabetes mortality in the country. And so Craig's been studying diabetes down there for a long time, focused on the genetics of diabetes, as well as interventions to improve diabetes rates in the community. But we went to Craig with the idea that we could study whether environmental exposures may be contributing to this devastating burden of diabetes in the county. And we also asked whether it's possible that underlying genetic risk may modify that relationship between arsenic and diabetes. So what do we know? So from the literature, we know from meta-analyses that arsenic exposure is associated with about a 70% increased risk of diabetes. So when you look across all the studies. So we took, at first blush, a cross-sectional analysis of this. So one snapshot in time across Craig's various cohorts to basically ask, how do urinary arsenic levels compare to diabetes in this population? And what we found is when these individuals were broken down by quartiles with the lowest quartile as the referent, those folks who are in the highest quartile of diabetes had a 5-fold increased risk of developing the disease. So high arsenic levels markedly associated with an increased risk of diabetes in this county. And again, under-recognized risk factor that we potentially could do something about. So this is cross-sectional stuff, and some of the problems with many of the environmental health studies is that this is sort of subject to confounding because you don't know like longitudinal relationships. And one of the great fortunes that we had is Craig was doing this longitudinal study of diabetes in Stark County. And so he had these almost 600 subjects that they were following for 3 years. And we had baseline measurements of arsenic and other metals at time 0. And so what we did was we basically then followed glucose control in these individuals over those 3 years. And what we found is that both arsenic as well as other metals were associated with an increased risk of deterioration in glycemic control. And so the question always is like, how do you translate this into something that matters to clinicians? So in looking at this, we basically decided to present the data as such. So what you can see here is months, and what we did that was really, I think, advantageous for us is in addition to having hemoglobin A1C and fasting glucose levels, we also had post-load glucose levels. So these individuals got 75 grams of glucose, we measured the sugar 2 hours later, and this is a more sensitive test for drivers of diabetes because it's provocative. It's testing the system to see how they respond. So based on the post-load glucose, what we found was those individuals with the highest levels of arsenic versus the lowest level of arsenic had a deterioration in their glycemic control that would predict the development of prediabetes nearly 2 years earlier as a result of their high arsenic exposure. And when you extend this out to the ADA diagnostic criteria for frank diabetes, they would develop diabetes 5 years earlier. So what we're seeing is an acceleration in that glycemic deterioration that means that individuals are developing prediabetes and diabetes earlier. And clinically, we know this is a problem because people are developing the micro and macrovascular complications of the disease when they get to the diagnosis of prediabetes. And the longer you have the disease, the more complications you're going to develop. And so here's an instance in which we think that that whole thing is accelerated in time because of their exposure. And what's interesting and that we didn't talk about earlier, but probably should have, is that the relationship's similar for nickel, which I don't have a really good explanation for, but there are other metals that these folks are exposed to as well that may be also associated with similar deterioration. Yeah. So in these individuals, what we're looking at is basically changing glycemic control over 3 years and then projecting out diabetes. But we know in the population, developing diabetes more and at a younger age as well. I don't have a direct answer to your question, though. These are just the characteristics of the cohort. I thought you were going to ask about that. But we know, like, when we think about diabetes, our clinical management isn't just about blood sugar, right? So what we're worried about are the microvascular complications of the disease. We're worried about the nephropathy, the neuropathy, the retinopathy, and the atherosclerotic cardiovascular disease and the heart failure that develops from this. And we know that the blood sugar is just one part of that story. So as part of this cohort, one of the other things we did is we looked at blood pressure changes. And so this is not published yet. We're currently working on this. But we also were able to show that those folks in the highest quartile of arsenic exposure also had worsening blood pressure over time. So their systolic blood pressure went up faster than those individuals who had lower levels of arsenic. And this is important as we start to extrapolate this out to the things that matter, right? So if you start thinking about Framingham risk scores and when are these people going to develop atherosclerotic cardiovascular disease or a myocardial infarction or a stroke, what you start to see is that these exposures are starting to hit multiple risk factors. And it's the summation of those risk factors that matter clinically. And so what I would argue is that it's important for us to consider all of these variables at the same time, because when we start to have conversations about attributable risk, we're often underestimating the impact of these things when we look at one outcome at a time, as opposed to looking at coordinate risk factors at the same time. And when we think about atherosclerotic cardiovascular disease, which, kills, 2/3 of people with diabetes about. I thought this study was pretty interesting. So this was a paper that you may have seen that came out in the New England Journal of Medicine this past year that looked at microplastics, another emerging source of environmental toxicants that really is just starting to get attention. So in this study of almost 300 individuals, they performed carotid endarterectomies for people with asymptomatic vascular disease and basically quantified the presence of micro and nanoplastics in this cohort. And what they saw is that those individuals who had these microplastics in their atherosclerotic plaques had a 4.5-fold higher risk of having a major adverse cardiovascular event compared to those people who did not have these microplastics. And so what's interesting about these microplastics is they may be a problem by themselves, but they are also vehicles of exposure for other toxicants as well. And so I'm telling you the story of arsenic, but it's important to recognize that what we're exposed to is a lot of different things doing a lot of different harms at the same time. And when we're looking at outcomes that matter to our patients, it's important to think about these things holistically. And that certainly goes to the development of diabetes as well. Because when we look at the spectrum of exposures, it's important to recognize that in most individuals, it's not going to be one chemical that does this. But each of us have thousands of different chemicals to which we're exposed. And so if we return to this paradigm of how we can think about glycemic deterioration, there are a bunch of different chemicals that have now been associated with impairments in insulin sensitivity through a variety of mechanisms that are well accepted by the metabolism literature. Similarly, there are a number of different chemicals that are associated with disruptions in insulin secretion. And this is based on both cell culture data, animal model, human epidemiologic studies, as well as actual clinical trials. And so when we start to think about how these things could interact, it may not be one chemical that does it. It may not be that Vacor example that I gave from the 1970s, but rather it might be death by 1,000 cuts. It might be some of these factors that modulate insulin sensitivity, coupled with other factors that affect insulin secretion, coupled with genetic risk, coupled with physical inactivity, together that sort of potentiate the development of diabetes. And this makes this difficult to study because there may not be one factor. But what it does underline is the fact that in each individual, there may be contributors here that we've been under-recognized. And this is just a paper that I just wanted to highlight because Matt was an author on this as well. This was a review of basically key characteristics of metabolism disrupting chemicals that just got published a couple months ago that kind of highlights how these different chemicals work through pathways that are commonly accepted to cause metabolic disease. Okay, so the other thing I kind of want to highlight is that one of the characteristics of diabetes is that it is not universally born across society, right? There are regions of the country with higher rates of diabetes. So the Deep South, Appalachia have higher rates of diabetes than other parts of the country. And this is sort of the latest map I got. I'm a little colorblind, so it's like problematic. But obesity and diabetes rates are higher in certain parts of the country. So this raises the question, are these higher rates potentially related to environmental exposures? And so this is, and when we think about these exposures and we think about the fact that you have multiple factors that may be affecting these pathways simultaneously, is there a way for us to look at environments holistically? Right? Where we can sort of, because, and the reason for this is when we think about the environments in which we live in, they're often good in many ways if they're good. And if they're bad, they're often bad for many different ways. And so how do these factors potentially interact with each other to contribute to the development of disease? So we had, while I was at the University of Chicago, I met somebody at UIC who is a postdoc at the Environmental Protection Agency that developed this environmental quality index. So this, and this was going to be the tool that we were going to use to kind of dig into this idea of, does the heterogeneity of diabetes risk across the country relate to the heterogeneity in environments across the country as well? So what the EQI is, it's a county level estimate of environmental quality That's a summation across 5 domains: air, water, land use, built environment, and socio-demographic factors, with each of those domains then composed of a number of different variables with that have been sort of developed in a way where higher EQI is a worse environment. So we basically, this is a county level variable, so it has problems, there's no doubt. Counties vary dramatically. The Chicago by itself like varies dramatically. But we decided to take this in and basically ask, does environmental quality matter to explain this variance in diabetes rates across the country? And so we looked at diabetes prevalence, and then we also looked at diabetes control. So using data from the Institute of Health Metrics and Equality, health metrics and evaluation, we basically looked at how well diabetes was controlled in different counties. So these are folks diagnosed with diabetes who have blood sugars that meet the threshold, right? So A1C less than 7, fasting glucose less than 126. And so what we found is that in those environments, in those counties that had worse overall environmental quality, that was associated with lower levels of diabetes control. In areas that had worse water quality, worse diabetes control. Poorer land use, less diabetes control. Worse built environments, less diabetes control. And the one that you would expect a priori, those folks who had worse sociodemographic factors, worse diabetes control. So again, this is sort of a county level estimate, but again, it points to this idea that environments do matter, and they matter not just for disease presentation, but also for disease control. We've done some additional studies on this to try to drill down into some of these variables, and some of these things are really cool. So I grew up in Chicago, so I'm interested in things like public transit, right? And so we can show, drilling down to public transit levels, that urban environments that have better public transit have higher or lower rates of diabetes. So this is not rocket science, right? Like people walk more, you have less diabetes. But what this underscores is that urban planning is a conscious environmental health choice, right? And so what this does is it opens up other areas of policy for interventions in order to stem the tide of the disease, okay? So when we think about environmental health, we need to be thinking about it across the spectrum of action because these variables matter. When we choose to put in a bus route, it matters for diabetes. Okay. So one of the things that we've known about diabetes for a long time that's really stuck in my craw is that diabetes is a disease of social injustice. So we have known for a long time that Black, Hispanic, Latinx, and indigenous populations have higher rates of diabetes. And when you look over time, despite the fact that we've known about these disparities, they have gotten worse over time. And it isn't just the disease itself, it's the complication from the disease. If you're poor, Black, or Hispanic in America, you're more likely to develop diabetic kidney disease, amputations, and eye disease. And if you're Black in America, you're more likely to have the worst complication of diabetes, which is mortality. Diabetes-related mortality is 2 times higher in Black America than it is in white America. This is a disease of health injustice that we need to do something about. And the question is it possible that some of these disparities are related to differences in exposure? So historically, we've attributed these differences to a variety of different factors. So education level, employment, food security, physical activity, and access to healthcare. All of these matter. All of these matter. But when you look at the health disparities research, environmental disparities aren't always part of that discussion. This is from a paper that did not mention it. I don't mean to call out anybody, but what I want to call out is the fact that we're not thinking about this adequately. So one of the things we did is we tried to look at this sort of holistically. And so my graduate student, Daniel Ruiz, who's now postdoc at Emory, what we did is we identified 5 classes of chemicals associated with diabetes in basic science studies, prospective epidemiologic studies, for which data was reported on exposure based on race, ethnicity, and income. And what we found in this analysis is that every time you look, those folks from communities of color and low-income communities often have higher exposures. And this holds for things like polychlorinated biphenyls. It holds for organochlorine pesticides. It holds for air pollution, which is enriched in communities that have higher rates of poverty and communities of color. And it holds for non-persistent pollutants as well, including bisphenols and phthalates. And I think one of the things that kind of sticks in my craw even a little bit more is that folks on emergency food assistance have higher levels of chemicals associated with diabetes like bisphenols and phthalates. Because when we give emergency food assistance, what we give are packaged foods, which we know are less ideal for metabolic health, right? And so we've looked at this again, we published this just a little while, end of 2023. What's important is that these exposure differences associate with health disparities across endocrinology. It isn't just diabetes, but it's obesity, it's liver disease, it's reproductive dysfunction, it's thyroid dysfunction, it's adrenal dysfunction. Like these diseases that are enriched in some of these populations are at least associated with some of these exposures. And again, I'm not saying that education and food access and healthcare don't matter, but I'm saying that these factors matter as well. And so the question is like, what's driving these disparities, right? And so we had the fortune in the paper in Diabetes Care to work with Carrie Ard, who's a sociologist at Ohio State University, to say, what drives these exposure differences? And it's not surprising. It is basically a century or more of racist policies. This is neighborhood segregation, the racialization of labor, grandfathering clauses for industrial sites that allow them to stick around in low-income communities longer. It's suburbanization and a lack of investment in urban cores. And so all of these things are factors that drive this. And there are more. This is not meant to be in a total list, but these are the factors that drive disease. And so when we think about what we're doing, we really need to think about how we can address these drivers if we're ever going to achieve health equity. I just want to mention this a little bit as well. So when we think about environments, I mentioned like a number of different things. We started with a single chemical. We talked about mixtures of chemicals that make up environmental quality. But endocrine disrupting chemicals and chemicals associated with diabetes risk are just one of many environmental threats. And we can't really talk about environmental threats without talking about climate change, even though we're not supposed to talk about climate change. So one of the things that we know is that plastics are a source of endocrine disruptors that are linked to metabolic dysfunction, but they're also produced from fossil fuels and therefore contribute to climate change. So we just contribute, and then so the question that we got asked in 2023 is, what are the relationships between climate change and diabetes? And so we just completed the scoping review that should be published any minute. I just finished the proofs yesterday, basically asking, what is the relationship between diabetes and climate change? In this scoping review, we identified basically 73 studies in humans that looked at climate-related factors, so specifically heat stress, cold stress, and natural disasters. We excluded pollution as part of this a priori, we were asked to. And what we showed is that individuals with diabetes exposed to extreme temperatures and natural disasters had worse blood sugar control, increased mortality, and greater healthcare utilization. And that's just where we are with the evidence right now. We need more data on this because there are key data gaps that we also identified. There are very few studies looking at the impacts of climate change on people with diabetes in low and middle income countries, including the countries projected to have the highest rates of the highest increases in diabetes rates over the course of the next few decades. There is very little data on the impact of climate change. And I would also argue very little data on endocrine disruptors and diabetes associated complications like retinopathy, nephropathy, and neuropathy. We need more data in those realms as well. And very few studies looking at pediatric, gestational, and type 1 diabetes. So huge data gaps here, but from the data that we know exists, there is reason to be concerned. And I think you would all know that a priori, right? So if you've got impaired kidney function from your diabetes, that puts you at increased risk of dehydration and kidney injury from heat stress, right? If you've got diabetic neuropathy and you have autonomic dysfunction, your ability to regulate your temperature is going to get screwed up and you're going to be more prone to heat and cold stress. So this is an area that I know we're not supposed to talk about, but we've got to talk about because a lot of people are going to suffer if we don't do anything about it. So what I'm arguing for really here is really centering the environment in diabetes health and disparities. And so when we consider things like the social and structural determinants of health, I think we need to take environmental exposures more seriously as part of this. Okay, so this is a shitty situation, but what are we going to do about it? So that's got to be the next step. And so what I would argue is that I think what we're looking at is action on multiple levels. And we're going to need to turn knowledge into action if we're going to stem the tide of this disease. And I also want to argue that medicine is a special case. And in medicine, I think we have special powers that we can use. So with regard to this, I think it is important that we acknowledge the fact that medical care exposes patients to endocrine disrupting chemicals. And I'm not saying medical care is bad. Modern medicine has saved countless lives and is critical for the quality of life that we enjoy today. That said, we are exposing our patients to bisphenols and phthalates and other chemicals, and we have to acknowledge that. That's part of the autonomy that we're supposed to be giving to our patients. Our ethical obligations are to proceed through principles of beneficence, non-maleficence, justice, and autonomy. And I would argue by lacking conversations about this, we are missing that. And so we've called on sort of various people and levels. So we need better policies, we need patients engaged, we need actions by manufacturers, and we need providers if we're really going to provide ethically sound, patient empowering quality. We're going to need policies. So we did this analysis a few years ago, basically asking the question, how does federal environmental policy address chemicals associated with diabetes? And the answer is they do not. Okay. There are some chemicals associated with diabetes that are regulated, but they're regulated for other reasons. They are not regulated for any relationship with diabetes. So when we start doing cost benefit analyses, we're missing all of this. We're missing all of this. And so then what we're left with is like, well, what about individual interventions? So what can we as physicians do to advise our patients to reduce their exposures? And so, there's some literature out there. I would argue that we need way more in regard to what we're going to do. But the problem is when you have a system problem and you try to make an individual solution, it is not the best approach. And the reason for this is that patients don't always have choices. They don't have the financial means to make those choices. Options aren't always known or clear. The interventions don't always work. There have been some classic interventions that have shown that the best intentions actually lead to increased exposures. The benefits are not well documented. And then the other aspect of this is that we know that there are sensitive windows of development, and the insult associated with the disease may occur years before the measurement takes place. So when we think about developmental programming, this becomes a problem. So what I would argue is that my patients and I don't always have the tools we need. And so this is really a tide. And if we're asking individuals to be the solution, we're like that person putting their finger in the dike. We are asking too much. For system problems, we need system solutions. And so as folks engaged in sort of the provision of medical care and the education of the next generation of providers, I think it's critical for us to make sure that this becomes part of education, that we are teaching our students, our residents, our fellows, and colleagues about the importance of environmental health, that we are engaging our professional organizations to get off their asses and do something about this, and that we're advocating for science-based policies that evaluate new chemicals for health effects and not just androgen, estrogen, and thyroid hormone disruption, that prioritizes human health and that eliminates the use of chemicals that increase morbidity and mortality. And as endocrinologists, we're like in a unique place in which our organization has been the leader here. The Endocrine Society has a special interest group on endocrine disrupting chemicals. I would encourage you to join the listserv. I would encourage you to be engaged in those activities. But I think it's also the case that we're at a time in which our advocacy has to be broader based than that. And so, thinking about our role as physicians is to protect the most vulnerable. And we need to step up. I mean, stepping up for environmental health is part of it. Stepping up for transgender health is part of it as well. And so these are just some organizations. We need to be active in our efforts. And what I would argue is pick your lane and do your thing. And collectively we'll do it all. So I just did this for you. You can take a picture of this as a reference, put it on your phones. I'll put it up again at the end if you want. Just a few people who might deserve a call. But I will say that collectively, these may be dark times, but we can do this. All right, this is my favorite slide I've made. We can do this. Together, we can do this. And so with that, I'm just going to close. I just want to thank my group. None of this is possible without them. I really don't know how to do anything. Like they do basically all the work. But I'm really grateful to all of them. Our funding we've been lucky to get, and certainly the participants of Stark County, the colleagues, collaborators, you for all your attention, the invitation to be here. It's absolutely great. And with that, the public flogging can begin. So thanks so much for your time.